Hey team, today the screencast is going to be talking about the process of anaerobic respiration. So, as you know, there are two different pathways, and the anaerobic respiration pathway occurs in the absence of oxygen. So there's no O2 involved in this process. And the initial substrate, or the initial piece that goes into this process, is the molecule glucose. Now, and there's one glucose that goes in. And now, as you know, glucose is a 6-carbon molecule, so let's start counting our carbons here. So, as you know from today's class, the first step in anaerobic cellular respiration is glycolysis. So this molecule is going to enter into step 1, which is glycolysis. And let's put glycolysis into a large box. This is going to be our box that represents the process of glycolysis. So this 6-carbon molecule enters, and the first thing that we need to do is put some energy into the system in order to essentially prime our glucose molecule in order to get out energy. So it's like making an investment that you would in the real world. You got to put in some money initially, so you buy some stocks or bonds, and then those grow over time. And similarly with glycolysis, we're going to invest some ATP at the beginning, and then we're going to get out more ATP later on in the payoff phase. So this is called the investment phase, and we are going to put in 2 ATP. So here we go, we're going to invest 2 ATP and get out 2 ADP. And then we're also going to invest some inorganic phosphate as well, so just some plain PI. And in this process of investing this phosphate, this ADP, and in this process of investing all this phosphate and this energy, our 6-carbon molecule is not only going to be phosphorylated, but it's also going to be split into 2 pieces. So if we look at it down here, this has now broken into 2 pieces right here. And if you look at our stoichiometry, what we're donating to the process, you'll see that we've added 4 total inorganic phosphates to our now 2 3-carbon molecules. So perhaps most importantly, that in the process of splitting this carbon into 2 pieces, it is oxidized. And you all now know that oxidation is loss of electrons, LEO. Loss of electrons is oxidation. And so in order for this molecule to be oxidized, it has to lose electrons. So it actually loses hydrogen and an electron. And what picks up those hydrogens and those electrons? Well, it's 2NAD+, a molecule very similar to NADP+ that you saw in photosynthesis. This, again, is a molecule that is a high-energy electron carrier. It's going to be important later on down the line in anaerobic respiration. But for now, you should know that this molecule is reduced. And this is actually, you can think of it as the key to glycolysis. If this molecule is not able to be reduced, then the oxygen, or the glucose cannot be oxidized, and the process can't be carried forward. So this sets us up for the payoff phase of glycolysis. Okay, I just left to go for a meeting. I apologize, I came back and I have no idea where I was. But the key here, you should know, is that this NAD plus comes in, it's reduced, meaning it takes electrons and hydrogen from the glucose molecule, or it's not glucose anymore, it's been split. And then these 2 3-carbon molecules are oxidized. And this is critical in the making or in the process of anaerobic respiration. And it's this process that allows, it primes these molecules to give up their phosphates to form ATP. So that essentially is the next part of this process. So we had the investment phase, and now we have the payoff phase, which actually the NAD plus and the NADH creation is part of the payoff phase. So I'm going to draw here. You see, this is part of the payoff. And so we spent 2 ATP initially, and now we're going to take 2 ADP, undo, and we are going to grab those, it's not 2 ADP, it's actually 4 ADP, and we're going to grab those phosphates on either side of those 3 carbon molecules to make 4 ATP. So then, once we've grabbed those phosphates from either side of those 3 carbon molecules, out pops at the end 2 3 carbon molecules, which are called pyruvates. So there you have it, 2 pyruvate pop out at the end, and they are not phosphorylated. So here I'm realizing something that I should have done earlier, is to label the steps in this process. So first the glucose molecule enters glycolysis. Next we have this investment phase, which is step 2 here. Then we have this inorganic phosphate being added, which you can think of as step 3. And then NAD plus is reduced, and these molecules are oxidized. That's step 4, and that prepares them to then have step 5, which I'm going to put a little star next to. And the reason for that is because this is a process called substrate level phosphorylation. And all this means is that the ADP is coming into direct contact with the molecule and is phosphorylated directly. The other type of phosphorylation is called oxidative phosphorylation, and you saw this oxidative phosphorylation process in our study of photosynthesis. Do you remember the buildup of the concentration gradient of hydrogen inside the thylakoid membrane, and then the release of those hydrogens through a molecule which is called ATP synthase? That was oxidative phosphorylation. So we draw a distinction between ATP created substrate level and ATP created through the oxidative phosphorylation pathway. Not because the final products are any different, but that they're just 2 different mechanisms of making ATP. So it's a term that you should be comfortable with and that you should know. Okay, so then let's move on to the next step in anaerobic respiration, which could be, as you know from today, 2 different things. It could be lactic acid fermentation or alcoholic fermentation, and we're going to start with lactic acid fermentation. So let's just write that here. So, in this process, we start with the output of glycolysis, which, as you know now, is where did that go? Oh, sorry, skipped a page. Is 2 pyruvates. again, ooh, that's too big. These are 3 carbon molecules. They don't have phosphates on them. 2 3 carbon molecules. And these are going to enter into our fermentation pathway. So let's make a box here that says fermentation. So, you should know that the only point in which we create ATP during anaerobic respiration, we've already passed that point. That was back here in the payoff phase, right here, when we took 4 ADP and we transformed them by phosphorylating them into 4 ATP through substrate level phosphorylation. But the key element here in glycolysis is we need this oxidation reduction reaction between these carbon molecules and this NAD plus making NADH to happen in order for this reaction to be pushed forward. And so in the process of fermentation, all we're doing here is recreating this NAD plus. So we won't make any more ATP, but we will oxidize this NADH to form NAD+, and I'm going to talk to you about how that happens. Okay, so we've got our 2 pyruvates, and we'll put them in our fermentation box. So let's select them right here, and paste them down below, and put this whole thing in a box. So essentially, all we have here is 2 NADH, 1 for each pyruvate, comes in and drops off hydrogen ions and electrons. NAD plus comes out. This molecule is oxidized, and these molecules, which are no longer pyruvate, are reduced. So we have reduction happening here. And then we can take these molecules. They are no longer pyruvate, they are lactates. And let's see if I can flip. Ooh, no. Okay, so I've changed their color here to represent the fact that these guys here are no longer pyruvates, but instead they're lactates. And lactate also can lose a hydrogen when it's in blood and the bloodstream in solution, so you can call it lactate or lactic acid. And again, this is what builds up in our muscles and causes us, when we feel the burn, this is what we're feeling. And then the key element here is that we have taken this NADH and regenerated it into NAD+. So now we have more NAD+ to run this glycolysis reaction. A key element also to this is that this lactic acid, this process of making the lactic acid is reversible, and we can actually regenerate pyruvate from lactic acid, and this would happen in the liver. So lactic acid is sent to the liver, metabolized, and turned back into pyruvate, and then stored and for a time when the body is not in oxygen debt, so oxygen is plentiful, and then we can actually stick this pyruvate back into the aerobic pathway, which where you see it will produce more ATP for the body. But under these conditions, making 2 ATP per 1 glucose is the best we can do. Okay, so this is lactic fermentation. The other type of fermentation that we need to talk about is alcoholic fermentation. So let's open up a new page. So again, in alcoholic fermentation, our beginning input is 2 pyruvate, and again, let's draw, oh, I spelled this wrong. This is fermentation, not fermentation. Let's draw fermentation, our fermentation box here. And unlike in lactic acid, which was just a simple rectangle, the fermentation pathway for alcoholic fermentation is going to look more like an hourglass. And we'll talk about why that is in a second. So, we've got our 2-pyruvate entering into our fermentation process. And so these guys jump in here, enter the pathway. Enzymes are going to be acting on them. We've got H2O, 2H2O, in the form of a liquid that's going to come in here and react with these pyruvate molecules, and we're going to get out 2 CO2. So we're going to grab carbon and take it off of our pyruvate. So now obviously this isn't pyruvate anymore, it's a different molecule, and that molecule is called acetaldehyde. And there are going to be 2 of them. So you can write 2 CET. 2-acetaldehyde, which, as you might expect, is a 2-carbon molecule. Remember, we just removed 2 CO2 from the pathway. So then it's this molecule that is going to be reduced by NADH. So we can draw in again here. We've got 2 NADH coming in here. It's going to drop off hydrogen ions, protons and electrons, and pop out 2 NAD+. So this NADH is oxidized. It loses electrons, and this acetaldehyde is reduced. It gains electrons. Okay, and then once those molecules have been reduced, they turn into ethanol. So from this we get out 2-ethanol. So this entire fermentation pathway, again, the whole goal is to recreate or recycle our NADH back into NAD+, so we can run glycolysis again and get out our 4 measly ATP in the payoff phase. And this all happens in the absence of oxygen. And so lastly, tomorrow in lab, we are going to be looking at this pathway here. We will see whether or not glycolysis and alcoholic fermentation have taken place by the presence or absence of CO2 molecules within our inverted test tubes. Okay, this was a really long one. I apologize. I'm going to end it right here. Pause.